Hello, my name is Jacqueline Torkelson. I am a fourth-year PhD candidate in the Department of Chemistry at the State University of New York College of Environmental Science and Forestry under Dr. Martise. And today I'm going to be talking to you about zooplankton fecal pellets as the primary driver of settling particles in reef systems. So while I am located in Syracuse, New York, this research did actually take place on a reef system. So first, I wanted to talk about settling particles in general. When you think about sediment and coral reefs, you probably think about smothering and high turbidity areas where the sediment could block out sunlight, and how just generally bad sediment and settling particles are for reefs. However, I wanted to flip that a little bit. So for this study, we wanted to think about low turbidity areas and how small amounts of sediment and settling particles could be beneficial for coral ecosystems. So for example, certain species of Scleractinia have been shown to ingest sediment, and they've actually been known to assimilate those nutrients into their tissues. This obviously varies depending on how heterotrophic a species is, along with their morphology and their general location. This can also change based on the zooxanthellae. If zooxanthellae are not present, for example, if the coral has been bleached, then feeding does become more important. In these low turbidity areas, while the sunlight can be blocked out, the zooxanthellae can actually photoacclimate and change their photosynthetic cells ever so slightly. Now, these settling particles are also home to a wide variety of lipids and other organic matter. These lipids can be an important source of nutrients for coral and fish and algae and other species generally associated with coral reefs. Now, these lipids can be used for multiple things. They can be used by us as scientists to track the source of organic matter. They can be also used for growth. So essential lipids being those that an organism cannot create by itself, and lipid meaning, in this case, fatty acids, sterols, and stanols. Fatty acids that most people are familiar with are omega-3 fatty acids that you would take as a supplement in fish oil, and the most common sterol people know tends to be cholesterol. And so the organic matter and lipids available in settling particles could possibly be a nutrient source that we hadn't possibly considered for these reef systems. This also goes into the role of zooplankton. So obviously this organic matter has to get from the surface down to the reef somehow. And in the open ocean, there's the biological pump, which has been widely studied, where at night zooplankton and fish come up and eat the phytoplankton, and then during the day they do return down to deeper depths. This can also produce sinking particles. And as you can see, the sinking particles get fewer and fewer as you get into deeper depths. Now, this is due to a wide variety of reasons. Some of it is due to just degradation generally for just existing in the ocean, and this is partially due to bacterial degradation. So the main thing here is to know that most of these systems that have been studied are in the open ocean, where they're thousands of meters deep, and you can collect water samples over hundreds of meters and see the transitions and see what's happening. However, our sediment traps were only about 2 to 6 meters deep, and so there's going to be quite a bit of changes to what you would expect from the open ocean, but it's still the same general idea. The main techniques that we used were looking at biomarkers and isotopes. So we use those biomarkers and isotopes to figure out where this organic matter was coming from, because obviously where the organic matter comes from is really important to figure out the nutrient content and how great something is going to be at providing nutrients. For example, it's probably better to eat something from a garden than something from the fast food chain. So that origin is really important. So we use these lipids as biological markers or biomarkers, as many of them are unique to certain species or classes. For example, on the top here we have dinosterol, which, if you can tell from the name, is from dinoflagellates. So that's the common name. There is a more fancy scientific name, but we're just going to go with dinosterol here. So this ring structure is generally what sterols and stanols look like. cholesterol looks pretty similar, which you probably have, floating around in your blood in various levels. Another example is a fatty acid. So on the bottom here, we have 25 omega-3 fatty acid. This one is very, very important for coral. If you're not very familiar with how to read fatty acids, just as a quick reminder, 20 tells you that there are 20 carbons total. The 5 tells you that there are 5 double bonds, or it's unsaturated 5 times. And the 3 tells you where that first double bond is. So in this case, from the end, we're on our 3rd carbon here, which is the omega-3. So this is an example of one of those omega-3 fatty acids that you get in your fish oil, which actually comes from other species and just is accumulated by the fish. Anyway, we also used isotopes. So we used nitrogen isotopes and carbon isotopes. We did bulk for both. We also did some compound-specific isotope analysis for carbon as well. Now, nitrogen can be used to indicate trophic level, while carbon isotopes can be used to indicate an ingestion pathway and possible sources of organic matter. And this becomes really important later on because some indicators of phytoplankton are the same as for zooplankton, but those carbon-13 isotope values are going to be what tells us which one is from which. So getting into our methods here, I apologize for the way that the pictures are laid out, but just to rearrange it so I could get my recording in here as well. At the top, we have a map of the continental United States, and the black box there is where the Florida Keys are located. So southern Florida, it is the largest barrier reef in the continental United States. Florida Keys Reef Tract, home to the Florida Keys National Marine Sanctuary. Our study took place in the upper Florida Keys, and so there is some slight influence from the Everglades, and if you're familiar with where Key Largo is, this is in the same general area. Our middle picture here shows us where the sites are generally pretty spread out. The sites ranged from about 2 kilometers from shore to 11 kilometers from shore. And then on the bottom is an example of one of our sediment traps using oh so fancy PVC pipe. So each one of them was left out between 7 and 11 days. After we got it back to the lab, we filtered each and used that filter to calculate organic matter and total inorganic matter, along with our bulk isotope values. And we then went on to do the specific lipid analysis. So for both of them, we derivatized the fatty acids and the sterols and stanols. So what I mean by derivatize is we needed to change it up a little bit and add on some protecting groups to make sure that we were able to read it properly using gas chromatography. So for the fatty acids, we methylated and ciliated them. For the sterols and stanols, which were our neutral lipids, we just ciliated them. We used those extracting methods by Teese et al., and then we measured using gas chromatography. So talking about sediment flux, we generally expected that it would decrease as you got further away from shore because there wouldn't be as much sediment coming from shore. However, we did notice that the offshore north was not quite what we expected. The most likely reason for this is this particular area where there is room between the keys, and sometimes water can flow through that area and bring a lot of sediment through it from the Everglades. So that's most likely why we had some differing sedimenting flux there as well. Getting into the results, there's a lot of bars here. I just wanted to highlight a couple of things, and in the next slide I'm going to break it down into what's really important. So really quick, the first thing you probably see here is like, oh yeah, cholesterol's pretty big, and the reason for that is it comes from so many sources that we can't really pinpoint exactly where it comes from. Another thing that I wanted to point out to you here is the presence of gorgosterols, which is a common indicator of gorgonians or coral. You can see that there really isn't a whole lot here, so there really isn't a whole lot of resuspension coming from the reef itself in order to have the coral really participate in the settling particles. Also, when we're talking about ocean systems and settling particles, we often talk about like bacteria, but we didn't really find much of a presence of bacteria here. So Odd chain fatty acids and these stanols here generally indicate bacteria, but they were in really, really small concentrations, which was very much of a surprise. Another thing that I really wanted to highlight was zooplankton. So while many of these lipids generally also indicate phytoplankton, based on the compound-specific isotope analysis, we figured out that all of these actually more closely matched zooplankton than phytoplankton, indicating that the phytoplankton were eaten by the zooplankton and it was hence repackaged as those fecal pellets. So on the top here we have essentially a culmination of all of the graphs. So again, the top is each one individually, the bottom is all of them averaged together. So you can see through the top that there really isn't much of a difference between each of the reefs. And on the bottom with all of the percentages, you can see that zooplankton really had the most influence on the composition of the reef, with phytoplankton coming in second. Again, bacteria did play a role, but really not as much as we expected. To summarize, the distance from the shore really minimally affected the sediment flux. It also really minimally affected the total composition of the sediment itself. Really not what we expected. Again, the lipid composition is directly influenced by zooplankton and their diet. So as the phytoplankton change, the zooplankton are obviously also going to change what they have in their diet. And while most of these systems are thought of primarily in these large ocean areas where you have thousands of meters of water column, you can see that it's really important. The transport of organic matter by zooplankton is still extremely important in water columns, but bacteria don't play quite as much of a role. I also wanted to quick mention how this will possibly change with climate change. We already know that organic matter remineralization is going to change. With ocean acidification, we expect that organic matter will remineralize at shallower depths. So we're not sure how this will affect the shorter water column compared to the larger water column. I also wanted to note that with climate change, there's probably going to be changes in the plankton dynamics, which will obviously also change settling particles in the area as well. Finally, I wanted to thank everyone that helped with this project, both Nurk and Diego Lerman, who were helpful in collecting these samples, Jessica for completing the fatty acid analysis. All of our funding was provided by NOAA and Nurk, and hopefully this paper will be submitted soon. It's in the last stages of editing. Hopefully it'll be submitted by the time that we have our virtual conference. I have my email here if you'd like to get in contact with me. I also have my Twitter if you would like to connect that way. Thank you for your time, and I look forward to talking with you more in a couple of weeks.